Okay, so welcome to Notes from the Lab, an ongoing series of conversations with experts around the world on the BCG vaccine. My name's Denise Faustman. I'm in Boston. I head the immunobiology labs, and we're really privileged here to take a glimpse at BCG from the perspective of Mihai Netea from the Netherlands, who's one of the global leaders of not only BCG work, but also the mechanism of how these bacterial interactions occur in the human as well as in lower animals. So let's get started. And maybe, Mihai, you want to talk about what currently is going on in your research lab today in the Netherlands.
Well, first of all, thank you very much for the invitation. It's a pleasure to be with you today. And indeed, in our group, we are studying a couple of projects, among which BCG, in trying to understand how do they modulate immune responses, and also what are the mechanisms behind it, and how is this working. In terms of some of the projects that we are very interested in, one major project is trying to understand in depth the molecular mechanisms that are responsible for the long-term changes in innate immune cells induced by the BCG. And of course, us and other groups have shown that epigenetic and metabolic modulation is very important for these processes. And we are trying to understand this at a more in-depth level. So one of the projects that we have worked in the last couple of years is trying to understand the relationship between metabolic processes in the cells, and lately we have focused especially on glycolysis and epigenetic changes, and the fact that an important increase in glycolysis with the release of lactate is determining chemical changes in the histones of the chromatin, especially lactylation, which is very important for containing the information of a previous stimulation. And histone lactylation seems to be an important molecular process that modulates epigenetic regulation of gene transcription in innate immune cells after BCG vaccination, and determining the long-term changes. That is one important project that we are doing. We are trying also to look at other chemical changes in the histones, other histone marks, in addition to lactylation, methylation, acetylation, and trying to understand how that interacts with the metabolic processes in the cells. That is one of the major projects on which we are working. Another major project in which we are very interested in is trying to understand how can we learn from BCG to build new drugs for new diseases. And here we collaborate with a group of Willem Mulder from the Technical University in Eindhoven, and we are building nanoparticles which are mimicking BCG that can be safely administered in an intravenous approach in order to treat especially cancer. We know that the intravesical BCG treatment in bladder cancer is one of the first and most effective forms of immunotherapy. Approximately 60–70% of the people treated with BCG intravesically for bladder cancer have a positive response to the therapy, which is higher actually than other forms of immunotherapy, such as checkpoint inhibitors. But unfortunately, because BCG is a live microorganism, we cannot give it intravenously, for example, in other types of cancer. So we are trying to mimic and build nanoparticles that look like BCG and behave immunologically like BCG, that can be used in the future, we hope in the near future, for the treatment of cancer. And that is one important area in which we are investing lots of effort.
Great, great. Do you want to tell the audience, because I bet even some of the MDs watching this long forgot the difference between innate and adaptive immunity. I'm sure they all had it the first year of medical school, and I'm sure they've all forgotten it. Let alone the poor lay people out there that are saying, okay, what's innate immunity? That would be really fun to share with everybody.
Of course. So when we defend ourselves against microorganisms, there are several layers of host defense that we employ to kill basically and eliminate pathogens. And in the first minutes and hours and days after an infection take place, there are specialized cells in the tissue, such as macrophages, that are activated, and they also recruit cells from the blood, especially neutrophils, that basically phagocytose, so eat and kill the microorganisms. To put it very simply, these are so-called myeloid immune cells, which are the cells which react in the very first hours and days after an infection, and they are very effective. And by far, the most infections are eliminated in this first phase of host defense. For example, when we scratch our skin and we get a very mild infection, or we inhale continuously every day pathogens from the environment. These are the cells that defend us immediately without even knowing us, without us getting sick, actually.
Mm-hmm.
They defend us against the pathogens. And we call that innate immunity because it's an innate process which is happening automatically. Even in a newborn, without learning too much, let's say, we can defend ourselves against infections. Now, in the minority of the cases when these mechanisms are not enough and the infection persists and get through this first layer of defense, then we need a more specialized host defense approach. And that is done by a second type of immune cells, which are called lymphoid immune cells, such as B lymphocytes and T lymphocytes. And they are recognizing very specifically the type of pathogen that are infecting us, and they mount a very specific immune response in the form of specific antibodies or specific T cell responses. They proliferate, and then they recognize specifically certain antigens, certain components on the surface of the pathogens, and they get activated to eliminate the pathogens. And this adaptive immune response, which is mediated by these T and B cells, by the lymphocytes, that needs a little bit more time. That needs 1 week to 2 weeks to get the full activation of these responses. The advantage, however, of these responses, in addition to being very specific, is that it can mount also very effective memory responses. After such an infection, some of these T and B cells that are activated, they transform themselves in what we call memory cells, memory T cells, memory B cells, and they can remember upon reinfection the previous pathogen, and they can be activated very quickly, so we can eliminate very quickly the second time the pathogen. That's why, for example, in case of certain infections, we get infected only once. We only have once in our lifetime varicella or measles or other types of infections. So this is a little bit in a very short summary, the difference between innate and adaptive immunity.
One of the things you discovered was innate immunity had memory. Do you want to talk about that? Because for years it was thought innate immunity was a one-hit wonder. So that's a pretty important observation.
Indeed, what it has been known for more than 50 years or so is that adaptive immune cells, T and B cells, have memory. And they build these memory lymphocytes, and they are responsible for an improved immune response after a first infection or after a vaccination. That's how we build our vaccines, by mimicking an infection, activating T and B cells to build memory. However, what we have observed is that there are changes, long-term functional changes that take place also in innate immune cells after an infection or after a vaccine. and that can lead also to an improved activation of innate immune responses. These processes are induced through different type of mechanisms, and they are induced through positing chemical changes in the chromatin, changes in DNA methylation, changes in the histones in the chromatin that induce the capacity of the innate immune cells to respond stronger a second time that they see an infection. And this is taking place in the peripheral innate immune cells, but also in the progenitors in the bone marrow. And that allows long-term accumulation of information, basically, of previous infections and improvement of the responses of innate immune cells in the future.
It's very interesting because I think most people who had immunology in med school, this kind of like went over their head, and it was not very interesting, but over the years it's become more and more interesting. So the next question, I don't have the answer to, but how did you get excited about BCG, or how did you pick BCG? Because there's a lot of organisms out there that interact with the host. So what was special about, did it pick you or did you pick it?
It was more by accident, to be very honest.
Okay.
We had a collaboration with one of my colleague, Reinhard van Krevel, working on TB and working also on trying to understand innate immune recognition of mycobacteria. And we were interested to see whether BCG vaccination can change the expression of pattern recognition receptors on immune cells. And we were doing a small proof of principle BCG vaccination trial to see what are the immune changes and how the cells respond differently to Mycobacterium tuberculosis. That's what BCG is meant for. But then we wanted to also to have a negative control to show that, well, those changes do not happen against other types of infections. And we had in our laboratory, because we were working also with fungal infections, we had Candida albicans. So we were stimulating before and after BCG vaccination the immune cells of the volunteers with both Mycobacterium tuberculosis and Candida albicans. And Candida albicans was meant as a negative control, basically. But to our surprise we were seeing that actually the innate immune response against Candida albicans was changed also after BCG vaccination, which should not have happened, basically. That was something that we didn't expect, and according to the immunology book should not be the case, because innate immune responses were supposed to be non-specific and not have any kind of memory, not change after a vaccination. And then we were observing that actually after BCG vaccination, innate immune cells such as monocytes, and later on we have seen also neutrophils, they respond better if they are challenged with Candida albicans, which should not have happened. So that's how we got interested in BCG, trying to understand how is that possible. And then we were looking in the old literature, and we found very old literature, even 100 years old, when BCG was introduced in the population, showing that a BCG vaccination was able to protect also against other types of infection, not only against BCG, against tuberculosis. And those information were present in the literature, but nobody could understand how is that possible. And then we got very interested to try to understand the mechanisms, the immunological mechanisms, the molecular mechanisms responsible for this old observation done with BCG.
Yeah, which is so much fun. I think it's great. When you write a paper, it forces you to go back and look at the literature. And sometimes it's just absolutely amazing. Oh my gosh, this now explains A, B, and C that was reported years ago. So I think this falls in that category. Well, as we finish up here talking BCG, do you want to predict where the field will be in the next 5 years? What do you hope to see? What do you hope to see? Yeah.
Well, there are several directions where I hope things will be investigated. So one of them is to fully harness these effects of BCG and other vaccines, because not only BCG, but also other vaccines have this type of heterologous protective effects. And
Mhm.
I'm talking
Mhm.
also about measles-containing vaccines such as
Yeah.
MMR, oral polio vaccine,
Mhm.
but also some of the new adjuvants, such as AS01, for example, adjuvanted vaccine seem to have these effects as well, such as Shingrix, for example. And I hope that people will do additional studies to understand these processes and use these already existing vaccines also for heterologous protection, so to improve this. That is one thing that I hope. Secondly, I also hope that using BCG, but also these other vaccines, we will learn which are the mechanisms that are important in these processes and what are the most effective ways of stimulating them.
Mhm.
So I hope also then that the people will also design a new type of vaccines which are meant to induce these heterologous effects. Because now we are just studying the vaccines that were built for other reasons. They were
Mhm.
built to increase an adaptive immune response. But I hope that we will learn from this and actually design in the future new vaccines that can induce innate immune memory responses and also eventually combine with adaptive immune responses so we build a new generation of vaccines that are much better and more effective.
Mhm. Not
And
just a single little antigen,
yes,
little mRNA vaccine that lasts a
yes,
short time, right? Yeah.
exactly. So I hope that we will learn for that. And the third one, I hope that we will learn from BCG and other vaccines what are important mechanisms in innate immune memory so we can treat other diseases as well.
Mhm.
As I mentioned, we are busy ourselves with finding new immunotherapy approaches for cancer. So
Mhm.
I think that there is a lot of potential there. So I hope that people will follow that as well. But at the same time, also to learn when these mechanisms are inappropriately activated and induce, for example, chronic inflammation. And we
Mhm.
are now identifying also diseases in which chronic inflammation based on innate immune memory processes are also important, and we are trying to design also therapies that decrease and inhibit these processes in case that they are inappropriately activated. So
Mhm.
I hope that all these three major areas will be followed, using properly what we already have,
Mhm.
designing new vaccines, but also learning from that for new diseases and designing new immunotherapies for other diseases such as cancer and inflammatory diseases. That's what I hope that it will happen in the next 5 to 10 years.
Well, the good news here as we finish up is a lot more people are working on BCG than I ever thought possible 10, 15 years ago. So I really enjoy seeing your papers come out, and I really enjoy seeing all the papers come out of how something so old can, catch the attention again of people where we didn't really understand it. And so we're going back to the basics. So
Yes.
today, I thank you so much for joining us. Notes from the lab. We hope it's not just for med students. We hope it's for the lay public so they can get a glimpse of a little bit more of the excitement of vaccine research. Thank you so much, Mihai.
Thank you as well.
Okay.